Hello, everyone. My name is Anastasia Danu. I'm from HELESC, the Hellenic Society for Crohn's and Colitis Patients. I'm very happy to welcome you in today's webinar, From Clinical Trials to Real Impact, the Future of Fibrosis Care. This session builds on last year's webinar, where we introduced the topic of fibrosis on IBD, explored both clinical and patient perspectives. Today, we will focus more on recent progress in clinical trials and what these developments mean in practice for patients. The FIBROTARGET project, coordinated by Professor Bram Verstockt, aims to improve how fibrosis is understood, diagnosed, and treated, including the development of less invasive approaches such as imaging. And before we begin, a few points. Please keep your microphones muted at all times unless you are called to speak. We are recording the webinar, so please keep your camera off unless you are speaking. And this helps maintain order on the screen. During the questions and answer session, you can raise your hand and once called, unmute yourself to ask your question, or write questions or comments in the chat at any time, and we will read them at the end. We will also like you to remind you that by registering, you have already agreed to the session being recorded. Anyone who does not agree can leave the workshop now. So let's get started. The first session is about clinical progress and imaging updates, and we will begin with an overview of the latest progress in fibrosis research, including how clinical trials are evolving and how success is being measured. We will hear about the role of imaging techniques such as MRI and ultrasound and how this may help reduce the need for more invasive procedures. And I would like now to invite Professor Bram Verstockt and Professor Alessandro Armucci for their presentation.
Thank you, Anastasia. And I will start giving a general introduction, and then Bram will take the lead explaining the core of the project, the last part of the FIBROTARGET project. If you can move the slide, let's go to the first one, just to let the community of patients to know what they are with. Probably they know, but it's very important to underline that we are in front of not only Crohn's disease, but also ulcerative colitis, two complex disease. They are complex in their pathogenesis. It's a mix of genetic environment, microbiota, and immune and non-immune cells that when the process starts at some point of the life of a person, then start and keep on going with inflammation. And the result is not a single disease, but as you can see in the middle, sorry about the picture, but there are extremely variable phenotype that may present under the name of inflammatory bowel disease. Still today, we are dealing with something that we are not able to stop in a substantial proportion of patients. That means progression of disease to intestinal damage. That is the onset of some extraintestinal manifestation. It is the risk to going into the hospital and to be admitted. That is the risk of surgery. That is still today the risk of colorectal cancer or other cancer. And we know that managing this disease costs money, but also costs quality of life. And this means a lot of indirect costs and a progressive disability. So the next one. Just to brainstorm on what are the main unmet needs in Crohn's disease, but there are so many that just I listed those ones that come to my mind. You see that we need some more precision medicine reliable markers. We need to better manage perianal fistulizing disease. We need to understand what does it mean, the control of residual symptoms when also inflammation looks to us controlled. We need to have a more durable and accessible treat-to-target care. But as you see, I have highlighted that we need to have effective therapies for fibrosis and stricture. And also probably we need to understand the process, and we need also to know how to diagnose and monitor this process. Because the next one, sorry, we know that fibrosis is a very complex process, and it comes from an interplay of many actors. We have immune and non-immune cells. We have bugs or bacterial product. We have myofibroblasts that, when the inflammation starts, they are activated and they transform in fibroblasts, and they start to produce extracellular matrix that accumulate in the muscularis propria at the beginning, then in the whole submucosa. And this is the starting of the process of fibrosis. And at the beginning is inflammation probably that leads also the fibrogenic process. But at some point, the fibrogenic process become independent and then keep on going if we are not able to stop or to slow slow it. So if you go to my last slide, this is a sort of go ahead. Sorry. Yeah, this is a sort of demonstration of what can happen in patients with Crohn's disease. This is a prospective cohort that was enrolled from the Danish database in the 2005, and then followed up for 7 years. And as you can see in the left part, this cartoon show that despite biological therapy, there is no control of disease in a substantial proportion of patients. Some patients change or location, and some other patients change their behavior. The majority of them, when they receive diagnosis, were pure inflammatory patients. Then a substantial proportion become stenotic patients. B2 means stricturing phenotype, and some other fistulizing phenotypes. And what happened at the end of the story in the first 7 years of disease? If you go to the right part of the slide, you see that those patients who changed their phenotype and they became from inflammatory to stenotic, or patients who were diagnosed stenotic since the beginning, were the patient with the higher risk of surgery. So this means that still today we are not able to control the fibrotic process in patients with Crohn's disease. So I think that Bram can start and take the lead, because he will start to explain what we are gonna to do with the FIBROTARGET clinical project.
Thank you, Alessandra, and good evening also on my behalf. So I think Professor Almuti illustrated it very nicely, that the unmet need is large and there is still a lot to be done. And we as a community, I would say, are super grateful that there is a lot of investment and development in fibrosis research. And you will hear later during this webinar more about new drugs being developed to really tackle the unmet needs. But while these new drugs are being developed, we wanted with the FIBROTARGET consortium to see, can we already try to understand a little bit better what currently approved therapies for Crohn's disease could mean to patients with stricturing Crohn's disease. And so this was exactly the reason why the CLEAR-UP study was designed. And so CLEAR-UP means Crohn's longitudinal evaluation of antifibrotic response to upadacitinib. And so this will be a study which will be conducted in 4 countries, the 4 countries who are basically representing academically the FIBROTARGET consortium. So with Belgium represented by me and Gianluca Mattioli with the Leuven side, and many other Belgian sites will participate. We have Spain with Hospital Clinic, with Ingrid Ordaz and Jordi Rimola and Azucena Salas. We have Alessandro Armuzzi from Milan, together with Maria Rescigno, Cristina Bezzio, and many other of the colleagues in Milan. And we have Marcus Neurath and Raja Atreya from Erlangen. So this will be a multi-country study in 60 patients with stricturing Crohn's disease with documented fibrosis, where we, in the coming months, will start, or where we will propose upadacitinib as a potential antifibrotic therapy. If you could click to the next one. So why upadacitinib? Why did we pick upadacitinib from all the drugs which are currently approved for Crohn's disease? The reason basically is that FIBROTARGET has many different aspects. And so now we are coming to the clinical trial, but in the previous years, we and many of our colleagues in the basic science field have worked within this consortium on trying to understand some of the biology. Why are people with Crohn's disease developing strictures and why are other people not developing strictures? And as you've already heard by Alessandro, we definitely haven't completely unraveled the full story yet, but what we have understood from all of these data analysis and from the biology perspective is that the JAK-STAT pathway seems to play a key role not only in inflammation, but presumably also in tissue remodeling and eventually also in fibrosis. And this was exactly the reason why we took the decision to use upadacitinib, which is basically approved for Crohn's disease, to explore further its anti-fibrotic properties. Next slide. So how will this trial basically look like? So we will have individuals with Crohn's with or without symptoms, but with a clear stricture on the MR enterography. And I'll come back to the MR in a minute. So these individuals will have at what we call baseline, so the start of the study, they will get an MR, they will have an ultrasound, and I'll come back to that as well. They have the endoscopy so that we can understand what is the impact from the stricture. We will do some blood tests, and all of these data, so the MR enterography, the ultrasound, the endoscopy, will all be analyzed together with one of the partners from the consortium. Motiland is a company who has developed a lot on interpreting these MR images, these ultrasound images, so that we can try to quantify to some degree, the amount of inflammation and the amount of fibrosis. And so we will start with upadacitinib on the regular dose, 45 milligrams, 1 pill a day for 12 weeks. This is exactly how someone out of the trial would also be treated with upadacitinib. And very early on, we will already do an ultrasound after 4 weeks to see, is this drug impacting on the stricture? We will repeat it at week 12, and at week 24, everyone will undergo another MR enterography, another ultrasound, and another endoscopy to see in these 6 months what has the drug done. So we will not give higher doses than normally. We will give the standard dose of upadacitinib, and then we will continue basically. And so the trial itself will basically run for 48 weeks, but also afterwards, we will still continue to follow up everyone who participates in the study so that we can see the evolution on the long term. Do these individuals with Crohn's disease have to undergo another surgery? Is there any endoscopic balloon dilatation needed, so that we might have some more data on the potential of really modifying, hopefully, the disease on the long term? Next slide. So how will we quantify? What does success mean? Of course, if someone has a stricture, as many of you might imagine, you might have a lot of abdominal pain, you might have a lot of cramps, you might lose weight because you cannot eat. So we will definitely capture in every patient the symptoms, including their food intake. But we do not exclusively want to limit this study to patients with Crohn's who have symptoms. If we know from the scan that there are strictures, also those patients will be welcomed in the study to see, even if you don't have symptoms, with this drug, can we basically, in a positive way, impact on the stricture? And so with this MR, these MR enterographies can give us a lot of information on the fibrosis and on the stricture itself. And so we will basically take advantage of the beautiful work the STAR Consortium has already done led by Florian Rieder and many others in the STAR consortium, who have defined some key radiological features of fibrosis in Crohn's disease. So we will look at how long is the stricture, and do we see that there is at least a 25% reduction in the first 6 months of upadacitinib. We will look at the diameter of the dilatation. If there is a stricture for your bowel, it becomes very, very hard to push the stools through, and as a consequence, the bowel will start to dilate. So if we can see that the dilation is going down, this is another sign on the MR that yes, the drug might be working. And the last part is also that we know that how thick the layers of the bowel wall altogether are, the bowel wall thickness, that this also can be a measure of the efficacy of the drug. So these three features we will capture together. And if we see after 6 months that 2 of these 3 MR parameters are improving, we will call this a first success. And of course, on top of that, we hope that those people who had symptoms already at the start of the study, that we will see that actually their symptoms will improve. So we will capture that in parallel. Next slide. You have already heard me talking about ultrasound. And so we realize that endoscopy and MR enterography are valuable techniques to give information. But the problem is, as many of you can imagine, that both a colonoscopy as an MR enterography puts a lot of burden to the patients. You need to do the bowel preparation for the tool. For the MR, you need to lay down for 45 minutes. You can hardly move. It's a lot of noise. So while this is still the standard, let's say, in the assessment of fibrosis today, we want to see, can we basically also capture the response or the potential antifibrotic effect to upadacitinib with an ultrasound? And again, based on what has been done brilliantly by the STAR consortium, we hope that we can basically capture a lot of the features with ultrasound, and you see an image there on the slide, which again can enable us to evaluate the bowel wall thickness, which can evaluate the dilation of the bowel, which can evaluate the length of the stricture, and on top, and this is a difference with some of the MR enterographies, with these ultrasounds, we can also see the function of the bowel. We can see if the bowel has challenges to push the stools through the stricture. So all of that will be assessed in the study. And the advantage of the ultrasound is that we can repeat it more often. So already at week 4 and at week 12, we can see, is there improvement? It would be impossible to ask for so many colonoscopies or so many MR enterographies, but with ultrasound, hopefully, we can see something very quick. And this is exactly the reason why it's so important that all of this, including the entire FIBROTARGET consortium, it's a joint effort with many together, and where there is a very important role also for IFCCA, because it's all about patients. What we do with this consortium is really trying to improve care and therapies for patients with stricturing Crohn's, and we cannot do that without having the patient voice here, not only in this webinar, but also while we are doing the trial, while we are creating the trial, so that we get all the feedback and the input, what really matters to patients, and that we can take that into consideration. So if we go to the next slide, this is my last slide. One more. Yes, so this is basically, these are 2 pictures from some of the meetings we've had, one 2 weeks ago in Barcelona and one a year ago in Milan. And I see that the resolution of the picture from the one in Milan is maybe not great, but you can at least recognize many different faces. And this is the key thing. This is not just a project led by a few people. This is really in partnership, and especially with a very strong voice also for the patients. So thanks a lot, not only to the people here tonight on the call, but especially to everyone involved, the radiologist, the pathologist, the basic scientist, and definitely also IFCCA for partnering with us in this together. It's a benefit for many of you, hopefully, not only now, but definitely also in the future. Thank you very much.
Thank you very much also, Professor Verstock and Professor Armuzzi. Thank you for the insights. As a patient with fibrotic Crohn's myself, I'm always excited to see steps on the right direction. And we now move to an update on the Stenova clinical trial from Agomab. And this is particularly relevant as effective treatment options for fibrosis remain limited, really limited, and new developments in this area are highly important for patients. So I would like to invite Tim van Kem to present.
Thank you, Anastasia, and thank you for the organizers for the invitation to present the results of the Stenova study today. And you can move to the next slide, please. So yeah, on behalf, you can click further. Yeah, on behalf of the investigators and in particular the patients that joined the study, I'm very honored to share with you the results today. If we move to the next slides, I'm actually very pleased that Dr. Armuzzi shared a bit the background of this complex complication of Crohn's disease, and despite the fact that we have a lot of anti-inflammatory therapies available, as highlighted also by Bram, and upadacitinib is one of them, there is still a progressive bowel damage accumulating over time, and that's the fibrosis that we obviously are targeting and discussing today. And moving to the next slide, as highlighted by Dr. Armuzzi, it's quite a complex process that is ongoing in the gut wall. But what is known for decades is that the TGF-beta pathway is one of the major drivers of fibrosis and the formation of fibrosis. And this is a pathway that has been described in literature and also has been investigated by many other companies and many other assets that block this pathway and has shown very promising results in organs other than the gut. But what we know is that this pathway is also upregulated in the gut and certainly in patients that have inflammatory bowel disease. There are multiple ways to block this pathway, and one of the most potent ways to block is through the receptor 1 proportion of the receptor, which is called ALK5. And that's also the target of ontonizertib. That's the drug that we are developing. The downside or the potential risk of this is that others have also attempted to block this through providing this drug systemically, and that has shown to be a risk for cardiac toxicity. This was shown in animals where you could see damage to the heart, and that's obviously something that has hampered further development of such agents. Now, if you move to the next slide, ontonizertib is actually designed to have only high exposure in the gut and avoid high systemic exposure, so that we avoid all these systemic liabilities of the class. So let me walk you through how it works. So the drug is taken orally, and it's traveling throughout your GI tract. We find roughly 10% back in feces, so we know that 90% is absorbed, and it's actually absorbed throughout your entire GI system, and it results in high exposure of the drug in your gut wall. And that's where it can have its activity. That's where, as presented by Dr. Armuzzi before, that's where the fibrotic process is occurring. But once the drug then moves into your bloodstream and is traveling through your liver, through the portal system, it's inactivated through a first-pass metabolism. So in your liver, there are a number of enzymes that help to degrade drugs, and what you then find back is inactive metabolites. So it means that there is a high exposure of the active drug only in the gut wall, where it has its positive effects, and you avoid any systemic safety effects because the exposure in the rest of your body is very low. So this concept obviously was tested previously in animals and also in healthy volunteers, but in the Stenova study, we wanted to see whether also in patients this drug could be safely administered, was tolerated well by the patients, but also that this gut restriction mechanism is maintained, because obviously patients that have inflammatory bowel disease have a slightly different function of their bowel than healthy volunteers. And you can move to the next slide. So the Stenova study was designed to in two parts. The first part was a double-blind part, which means that patients or physicians don't know what they are receiving, but patients were randomized to three groups. A high-dose group of ontonizertib, 200 milligram twice a day, a low dose, which is 100 milligram once a day, and a placebo group. And all of these patients needed to have an ileal stricture or a stricture or stenosis in their small bowel, and they need to have symptoms like explained by Dr. Almuzzi, pain after eating or changes in their diet, and they could be on standard of care. I think that's important. As shown, patients have Crohn's disease, they are allowed to stay on their standard of care, and then we add ontonizumab on top of that. And these patients were treated for 12 weeks, and we also assessed, as presented by Bram, we looked at endoscopy, we looked at MRE, but also how the symptoms of these patients resolved. This was a global study, a study performed with 52 sites, we enrolled, or we were able to enroll 103 patients, and I will walk you through the results of Part A. All the patients that had completed Part A could roll over into an open label part where patients were offered the high dose for a period of another 48 weeks, and that part is actually still ongoing. So moving to the next slides, it was important to learn about the patient population. This is the first of its kind study in patients that have stricturing disease. And it was enlightening to see that in this particular study, compared to other studies in Crohn's disease, that these patients have a very long disease history. And that also is something that the progressive bowel damage is something that builds up over time. So it's consistent with this long disease history. What we also saw is that a lot of patients had already had prior surgery. So there is clearly a high reoccurrence rate of a stricture even following an initial surgery. And as these patients, most of them, 75%, were on advanced therapies, we still saw that there was some mild luminal disease activity, but overall the systemic inflammation was well controlled, as you can see by the low CRP levels. So I think what to keep in mind is long disease history, very severe and refractory population that we are trying to treat with ontonizumab. The first question that we have, and that's on the next slide, is obviously whether the drug was safe and well tolerated. And in this study, indeed, we showed that ontonizumab had a favorable safety profile. This was confirmed by the high completion rate, so 90% of the patients completed the first part. We also saw that when we looked into adverse events, that this was well balanced. We didn't see a difference between whether you were on placebo or on active drug. And we specifically zoomed in on some of the class liabilities that were described. We had extensive monitoring of heart function, half-valve, and we didn't see any toxicity in any of these parameters. Also looking at patients' labs, liver function, etc., we saw actually that ontonizumab was devoid of any of these liabilities. Throughout the study, as it was a first of its kind study, there is an independent data safety monitoring board that assesses the safety, and there also advises to continue as planned. The second objective of the study on the next slide is to confirm whether the gut restriction mechanism also works in people that have IBD. And very schematically, if you look on the left part of the slide, you can see at the bottom these blue lines, and that is actually the low level of ontonizumab that is circulating in your blood, whereas the gray lines are representing the inactive metabolite that is formed once the liver has cleared the drug. So what it confirms to us is that the liver first pass metabolism is working, but also that the drug was absorbed, because otherwise we would not be able to find back this metabolite. And as you can see, depicted by the blue arrow, we stay far from any biologically relevant levels of ontonizumab, so we are devoid of these systemic liabilities. Data that is not on the slide, but that we also collected, is that we collected biopsies of patients at the site of the stricture, and there we measure really high concentrations of the drug, so we know that the drug is also present in the tissue and the site of interest. Then the last part of the study on the next slide is that we, as shown by, presented by Bram, there are multiple ways to assess disease activity in patients, and the most common way is endoscopy. And we started from looking at what we do in routine clinical care. We there assess the disease activity to a score called SEACD, the Simple Endoscopy Index, which assesses the level of inflammation, but also in particular looks at the narrowing component of a stricture, whether the stricture is passable or not. If we look at the standard definitions that are being used, which is, for instance, a 50% decrease of the score, showing an improvement, then we see that at the high dose group, 30% or almost 30% of the patients showed an additional response on top of their background therapy, compared to 11% in both the placebo and 100 milligram group. The same for remission, which is a harder criterion. It means that really there is only residual disease activity left. Also here you see that at the high dose group, almost 24% of the patients showed an improvement on top of their standard of care, compared to 8% and 6% of the respective placebo and 100 milligram groups. Moving to the next slide, as said, the SACD score is also able to assess the narrowing, so the passability of a stricture. And for that, we did a post-hoc analysis where we zoomed in on patients that had a non-passable stricture at baseline, so a stricture that could not be passed with a scope. And you see that there are slight imbalances in the number of patients that had these non-passable strictures. But what is also clear is that at the high dose group, again, we see that roughly 30% of the patients had a stricture that opened up after 12 weeks of treatment, whereas only 7% approximately in the placebo and in the 100 milligram group showed the same. Which led us to zoom in a bit deeper on the stricture morphology on the next slide. And there, the modality that we used in our trial, and indeed thanks to the work from the STAR Consortium and also FIBROTARGET, we were able to start looking into certain parameters that define stricture morphology. Although this is a very early readout because fibrosis is a very slow process, we start to see trends that point towards an improvement, and in particular for stricture length. What we see on the left is that there is a progression of stricture length still ongoing in patients that receive active treatment, but it's far more, it's faster, or it progresses more on placebo compared to active therapy. So also here we start to see signs of clinical efficacy only in a short or after a short period of treatment. When we look at the other relevant parameters like associated diameter, the dilation diameter or the bow wall thickness, then also here we see trends that move in the same direction, but also we have to be careful here. There is still quite some variability in this data. So maybe to conclude on the last slide, I think we fully echo that this fibrosis Crohn's disease remains an unmet need in stricturing disease, and that in the SENOVA study we evaluated ontonizertib as a first potential future therapy for FICD patients. The drug was well tolerated. We didn't see any safety signals. We confirmed that it was gut restricted, and we start to see first signs of improvement when we assess the patient's disease activity and narrowing component of the stricture through endoscopy or MRE. So this has led us to start thinking about the next program that will start later this year. Thanks.
Nastya, you are muted.
I'm sorry. Thank you very much, Tim. It was very interesting. And now we move forward to IFCCA's role in clinical trials and patient involvement. And I would like to ask Vaso Vakufci and Roberto Saldana to start their session.
Okay. Thanks. I'm waiting for Vaso.
I was muted.
Okay, sorry. Sorry.
I was speaking alone. Okay, sorry about it. Good afternoon, everyone, and thank you for being here. I'm very pleased to join you today, representing IFCCA, together with my colleague Roberto. Before we talk about fibrosis, treatments, or clinical outcomes, I would like to take a step back and look at how we develop solutions for patients with first place. Because this is where everything begins. As you can see on this slide, patient involvement is not a single moment. It is a continuous process across all the entire research and development pathway. From setting research priorities, to study design, to trial conduct, and even after approval through dissemination and health technology assessment. And this is exactly where IFCCA plays a role. At IFCCA, we are actively contributing across all the stages. We help prioritize by bringing forward real-world unmet needs from the global IBD community. We contribute to study design, ensuring protocols reflect what is feasible and what is really meaningful to those living with IBD. We support trial conduct by improving recruitment or retention and improving the patient experience during the trial. And we are present in post-study phases, translating results, informing policy discussion, and mainly contributing to HTA process where we have a long story. So this slide for us is not just a framework, it is a proof that meaningful patient involvement is possible. And IFCCA is already demonstrating how it can be done across this entire pathway. Because if we want innovation to translate into real impact for those living with IBD, those living with IBD must be at the center. And now I'm passing the floor to Roberto for the next phase.
Thanks. In fact, we are doing all of this listening to the patient. Bran already told us that behind all of these definitions, figures, and trials, there are real people. People with IBD are suffering, and we are listening to them. And they are really highlighting the importance of keep researching, because they are still in need. They are living in pain, living in fear, because of the scenarios they are dealing with. For example, as a consequence of getting a delayed diagnosis, a permanent damage, that even when they are able to scale up their drugs, their treatment, sometimes it's not even possible of getting any advantage of that. And this is because of fibrosis in many cases, because of when the damage is there is no going back. We can't reverse that damage, and we need to work on that. And this is why we are here and doing what we are already proposing, trying to avoid this eternal cycle where we are afraid of surgeries, afraid of risking our life, our quality of life, taking decision, permanent decision, modifying our careers, our roles in our families, group of friends, even in our societies. So listening to the patient, we really understand that we have some symptoms like abdominal pain, among others, and that information is what we are using to propose the endpoints for the trials, the PROs, the patient report outcome, that are really meaningful for us, and the information we need to track during the trial, so that we can be sure that the result of the trial is offering us what we really expected from innovation, getting our life back, for example. Next, please. And we are, at the same time, still working on other initiatives that are also facilitating us the context of how to conduct these trials, because we can have the proposal, we can have a promising drug, we can have the best endpoints, but we also need to be able to transmit that information to the potential participant so that they can consider this participation. And sometimes patients are telling us that trials are not friendly because of we are not adapting the trial, the frequency of the visit, the procedures, the invasive procedures to their preferences or to their real life, because location of resident is affecting the participation of the trial. Sometimes is the number of time the questionnaires to submit are taken. So we need to listen to the patients at the same time that we are developing things for them, for us. And we need to do it because we need to accelerate the trials, the recruitment, so that we can get the result as soon as possible and understand if they are successful to understand if the final drugs, when everything is happened as expected, can reach the market and improve the life of those who are waiting for new solutions. Next.
And so, what do patients really expect from a clinical trial? For patients, a clinical trial is never just a protocol, it's a real-life decision, often made at a very vulnerable moment of their life. And this is exactly why tools like this IFCCA guidebook for patients to review clinical trial protocols are so important. They translate clinical trials into something patients can easily and actually understand, evaluate, and question. Through IFCCA, we actively, of course, support this kind of work, ensuring that patients are not passive participants in a clinical trial, but also informed reviewers and contributors to the trial. And when a patient looks at the trial, they don't start with endpoints. They often start with very human questions like, is this trial designed for people like me? Are the inclusion criteria realistic, or they do exclude too many real-world patients? How burdensome is the participation? How many visits, how many tests, how invasive procedures? Because from a patient perspective, success is not only a clinical remission, it is avoiding surgeries, it is reducing pain and urgency, managing fatigue, which is a main problem for many IBD patients, maintaining social and emotional well-being. And as you can see here, these dimensions, efficacy, safety, and quality of life, are all part of how patients assess a trial. So this changes the perspective completely, and this is where IFCCA plays a key role. We work to ensure that all of these expectations are not just expressed, but integrated into how trials are designed and evaluated. Because if a trial does not work for those living with IBD in their real lives, then it will never be succeed, no matter how strong the evidence or the data are, because they have failed the main points of what is really meaningful to patients. I don't know if Roberto wants to add something about the guidebook, but I think that you can search it in our site, and you can also use it in your daily life, even if you are a practitioner who has to conduct a clinical trial, or if you are a person living with IBD, you can go through this guide and see how it can help you. Thank you so much.
Thank you very much, Roberto and Vaso. And now we move forward to questions. Anyone who has a question can write in the chat. I have a few questions, and I would like to start from the end, from the IFCCA's role. Okay, we have a question on the chat, and so let me ask this question. Since you are measuring length and thickness, how do you actually quantify if the change is true remodeling of the scar tissue versus just a reduction in leukocyte infiltration or swelling? I'm guessing this goes to Agomab and the Clear Up participants.
I assume it's indeed about the clinical trials, and Tim, please chime in and add. So it's an excellent question, and we should be very clear that inflammation and fibrosis, they are intertwined, and that's also what we see on histology when a patient has to undergo surgery and you look at the resection specimen, there is never only fibrosis and there is never only inflammation. So it will definitely be a mixture. But if you then see also on the endoscopy, for instance, that the stricture is not possible, this is an additional argument to say this is presumably already more fibrotic and part of the FIBROTARGET project, and I didn't go into too much detail, is actually really trying to understand as much as possible basically that complementarity and where the differences are. So Jordi Rimula, who is a radiologist in Barcelona and who has been leading a lot of the imaging work in the consortium, has actually really been working on a model where we basically have MR images from patients who subsequently underwent surgery. And so what he tried to do is trying to link the degree of inflammation or the degree of fibrosis with the features on MR angiography. And so this is still in a validation setting at the moment. But what we will do, exactly for the reason pointed out in the question, is that the clear-up study will actually be reanalyzed afterwards if we can validate that actually the model can separate the two to some degree from each other. But maybe Tim, you can also comment from your end as well.
No, I think indeed it's an excellent question. I think it was, as you highlighted, Bram, in the clear-up study, I think there is a lot of efforts by different academia, FIBROTARGET, STAR, investigating how to use these tools and what a meaningful change in some of these features actually means. So is a 25% reduction of stricture length meaningful for patients? And I think that's an ongoing effort. I don't think today there is a clear answer forward. So I think us, but certainly the academia, are using all the tools that are available to assess how the patient indeed is responding to a certain therapy. And obviously in the end, we all have the same goal, and that's to make the patient feel better and better function and have a better functioning bowel. So yes, I think very good question. I don't think we have the answers today. I think that's why we are joining forces to learn about this. And that's why the academia and the partners are trying to answer all of these questions.
Thank you very much, both of you. Mrs. Angela Clark has a question. The stage is yours.
Hi, can you hear me? Yes, hi.
Yes.
Yes, I'm Angela Clark. I'm president of Propel Cure for Crohn's disease, which is a US-based nonprofit raising funds for research for Crohn's disease. And I had a question about the Stenova trial for Mr. Van Kaim. Do some of the participants in the trial, have they had anastomotic strictures? Is that one thing that's being targeted? And secondly, is this potential drug seen as something to take as long as one has strictures, or something to stay on indefinitely at this point?
Yeah, no, thanks, Angela. Good question. So yes, indeed, patients that were allowed to have anastomotic strictures. So we investigated both populations or both type of strictures, naive and anastomotic. So yes. Secondly, yes, I think indeed the idea is that Crohn's disease is a chronic indication. So the idea is indeed that this is a chronic treatment. Obviously, that still has to be evaluated. This was a first study. We're not there yet to really understand the long-term safety, tolerability, but also the treatment and dosing regimens. That's part of the drug development process. So, but indeed, it's probably going to be a chronic treatment.
Tim, if I can add something.
Of course.
Yeah. Because I say to Angela that all of these patients had been operated in the past. If you remember, in the baseline characteristic, the duration of disease was a mean of 16 years. And to be a stenotic patient, you know that after 16 years, the risk to have undergone to surgery is almost, I don't want to say 100% in this category of patient, but high percentage. So very tough patient, also difficult to find for this kind of trial because they are not simple, with some kind of exclusion criteria to find and also to convince to participate to something that is really new, actually, really new. It's not another anti-inflammatory drug, but is a new concept to treat Crohn's disease.
I wanted to ask, if I may, just one more thing about the exclusion criteria of the trial, because it currently, the Stenova trial, it currently excludes patients who've had extensive resections, and also I believe ones who've had balloon dilations closer than 6 months apart. So that probably excludes a lot of patients who could benefit, even if their disease is under control. So I didn't know if any of that criteria would be loosened up later in the process. I imagine so, but I didn't know.
Yeah, also excellent question. I think when as a drug developer you want to evaluate the safety of your drug for the first time, you have to be much more careful than in, let's say, routine clinical practice. And I will leave the floor also to Bram and Dr. Amouzi to comment. But yeah, we have to be a bit more conservative. So patients that have undergone an EBD, let's say a couple of weeks ago, then there might still be some response to that. And that's obviously something that in your clinical trial you want to avoid. You don't want to face patients that are maybe having some wound healing process ongoing and that they are in your first trial, so we exclude them. Also, patients that have extensive bowel surgery and maybe only short bowel syndrome or something, there we don't really know yet how the drug is going to be absorbed because that's sometimes impacted. So also there, that's a patient population that at that point in time maybe is not the ideal candidate for such a first study. So yes, it's out of precautionary measures that we exclude that type of patients, and that could be modified when there is more clinical data available over time.
And can I ask a question relevant to this one? How about patients who were already in some kind of medication for also other autoimmune diseases? Many patients have symptoms of Crohn's-induced arthritis, psoriasis, rheumatoid arthritis, and they are already in some biologic drug that covers both Crohn's disease and the exterior symptoms. Can they take them together, or it has to be monotherapy with the sorry, I cannot.
Anthony Sertif.
They can take together. They can take
Exactly.
together.
Yeah.
Yes.
Okay, and it's not an exclusion criteria to have underlying conditions. And what about patients who have cardiological problems? Because you mentioned and you insisted on cardiac toxicity. How about people who have hypertension, POTS, other cardiological problems? Are they excluded from the trial?
It's obviously always a medical assessment, a medical judgment. So I will leave the floor to both physicians on the call. What we particularly looked at is whether there is any known damage, obviously, to the heart before patients are enrolled to the study. So if there is any pre-existing condition, then these patients at this stage would be excluded because they could potentially be at high risk. And we screen the patients and monitor the patients that don't have that throughout the study to see whether we don't induce anything. And luckily we didn't. And that's also something that in the development of ontonizumab, we have extensively studied in animals before moving into patients, and we never seen this. This is a class effect that was described for others at much higher dose levels than what we see with ontonizumab. But there is obviously a courtesy and a responsibility to monitor that when you develop new drugs.
Yeah, at this stage of the development, I think that they have to be as much as possible careful, but these do not exclude that if no signal will come out in the future and the development will go further, then maybe that also some patient with some potential cardiac problem could be suitable to be enrolled. But today we do not know at all, actually.
Correct. Yeah.
If there is no other question right now, I have a question for the upadacitinib trial. Where I live, I'm from Greece. Upadacitinib is not a first treatment drug. People take it as a third, second choice. So is the trial going to have to include the participants who are in their second biologic, their third biologic, or is going to use first-time treatment?
No, it's an excellent question, Nastya, and so we wanted to be as inclusive as possible to mimic real world, and of course this has potential risks in a way that the effect might be limited by patients who unfortunately already have had many therapies which didn't work. But again, to really study the potential of JAK inhibition in structuring Crohn's, we did not put any restrictions on number of therapies. So, for instance, patients being enrolled in the study in Germany can receive first line, as it's available in Germany first line. It's not in Italy, it's not in Spain, it's not in Belgium. So we will basically welcome everyone who has not received a JAK inhibitor before entering the trial.
Mm-hmm. Can we move to IFCCA and ask the €1,000,000 question? The main concern of every patient, including me, in different levels, is the side effects. How can we educate? What's the plan to educate patients for drug side effects, not just to make them not be their major concerns? We have patients who smoke, drink, eat greasy foods, drive recklessly, and yet when they ask what's your most major concern, is side effects from the drug. I don't know, anyone who wants can answer from the participants. How can we tell patients that side effects is not the major concern they should have?
Patient first. I would like to know the opinion from the patient side, that we can give our opinion, but first you, please.
And, okay, I can do it. I'd say that this is a pending task. There are so many ways to do it. Like the clinical trial information, patients are also afraid of the clinical trials. Maybe because there is a lack of information, and this is the same with the side effects. I'd say that we are more worried about the benefits of the drugs. And when we have already checked the benefit, we can make the trade-off. So that is, we can speak about side effect without taking into account the benefits, because this is how we decide things. For example, being adherent to the treatment. However, we are also missing some opportunity to speak about how we produce and develop the drugs, how the clinical trials are so safe, because we are scaling the drugs according to how we are generating the information, even telling to the patient if the drug is really innovative, if they have been used for other conditions. When people just only heard from us that it's not an innovative drug, that is something that has been proven in other conditions, people feel better, because maybe it's not effective for them, but at least it's safer than they expected. And with how we are also able to participate in pharmacovigilance, there is a room for improvement there, because there is a lot of information to explain how we can participate, and also check the information, available information from official websites. So we can also check and monitor how we are producing evidence about the safety profile of the drugs, and also being able to participate. So there is a lot of opportunities, a lot of ways at the individual, also at the group level, but maybe we are so cautious to speak about side effects. We also need to think about it. We as a professional, as a patient organization, because we are maybe worried to scare the IBD patients. And this is something we need to consider.
Thank you, Roberto. And we have 1 minute left. So I would like to ask everyone if they just want to make a short, really short comment about the people at the to the beginning and the end of the target groups, people who are 18 to 20 and people who are 65 plus. They are usually excluded from clinical trials, but they still take the drugs. I would like to know if they are included in these clinical trials, and if there is any plan to educate those patients in order to participate more.
Just very briefly from the ECHO side, since I'm working also for ECHO, we just had a sort of chat with the European Medicine Agency, asking them to stimulate industries to include adolescents in adult trial from 16 years old, because some trial have already started, and this could be a big achievement if any new phase 3 trial that usually allow the drug to be approved may include also some kind of adolescent person. From older people it depends. It's more difficult because if you are 70 years old but you are fit, you can rely on the result on a 60 years old or 50 years old patient that have undergone to a clinical trial. There is really no difference at all. The problem is that if there are comorbidity, and then it becomes more complicated to be enrolled into a trial, because side effect could be really dangerous. So it's a sort of risk-benefit balance. And for older people that have no big comorbidity, I think that they can rely really on the result on a 20 years less old patient that have participated to the trial. This is my opinion, sorry. I don't know if Bram and team can also agree on that.
Completely agree, Alessandro.
Mm-hmm. I completely agree, and to make it more specific to the CLEAR-up study we proposed, so there, because we are using a drug which is approved, we have to stick to the label of the drug. So that means that we cannot recruit people below 18 years of age, because the upadacitinib is not approved there. And basically the same applies there when it comes to JAK inhibition and the PrEP recommendations we have, both in Europe as also from FDA. We need to stick to the label, so we cannot deviate from there, and therefore we can also not go beyond a specific age, because of course we want to move the field and we want to improve patients as much as possible, but we also should be cautious of safety, obviously.
Also, from the patient perspective, I will not repeat what already said about people aged 65 and above. Of course, if we speak about people living with IBD under the age of 18, we speak about pediatric clinical trials and not adult clinical trials, so it's another field. But for those who live with IBD and they are between 18 and 30, we have recently run a survey at IFCCA, and we saw what really mattered to patients at these ages, and the results were very interesting. You can find it on our site, or we can send you right after. And also, when it comes to a clinical trial at these ages, they are more reluctant sometimes because they have not tried everything. They still have some options, so a little portion of these patients really accepts to participate at a clinical trial, but we have to focus on this age group also, and to educate them that a clinical trial is not, sometimes it's not when you are at the end that you don't have any option, but sometimes maybe it will be a better medicine for your case. Thank you.
Well, we took 4 more minutes of your time, so I think we can call it a success. Thank you everyone for your participation. Thank you everyone for the questions. Thank you to all the speakers for the very interesting, as a fibrotic patient myself, I found the webinar really, really interesting and really, really hopeful for the future of fibrotic patients. I think this, if I may make a comment, this is a war we're going to win, one battle at a time. Thank you very much. Have a nice evening, and see you at the next webinar.
Thank you. Thank you all.
Thank you.
Bye-bye.
Thank you.
Bye-bye.
Bye.
Bye.
Bye.